Welcome to Monobiology. Today we're going to learn a little bit about phylogeny and systematics. And for you AP Bio folks, this goes along directly with the booklet that you have on the same said subject, phylogeny and systematics. So what is phylogeny? It's an interesting name, phylogeny. Phylogeny. Phylogeny is the evolutionary history of a species or group of related species. So it could be one particular species, or it could be a group of related species. Let me give you an example of that. It could be just humans and their direct ancestors, or it could be the entire group of apes that are technically related to humans. That would be, so it could be either of those. Now, information used to construct a phylogeny can include both the fossil record, but also molecular comparisons. And those molecular comparisons, there should be some commas in here, let me fix that, would include DNA, RNA, and protein. And they can be used for those molecular comparisons like I was saying. Okay, so you can compare DNA, RNA, or protein, or all three, to try and determine how closely related individual species are. Let's talk about systematics now. Systematics is the study of biological diversity and evolutionary context. Systematics encompasses and includes taxonomy, and if you recall, taxonomy is the science of naming and classifying organisms. Modern phylogenetic systematics is equal to the classification of organisms based on their evolutionary history. So it kind of takes taxonomy to another level. Right? Taxonomy itself originally started off as being a system of classification based on common characteristics. This takes it to another level. So we don't just look at common characteristics, we look at how closely related the organisms are, and if they're more closely related, then we put them in the closer related groups in terms of our taxonomic sequence. Okay, so here we have what's called a cladogram. And a cladogram is useful. There's a few things I'm going to tell you a little bit about, a few things I'm going to get you to write down about a cladogram. A cladogram, first of all, is a phylogenetic diagram based on cladistics. Okay, now what the heck are cladistics? Cladistics are a form of taxonomy based on shared characteristics. I'll say that again. Cladistics are a form of taxonomy based on shared characteristics that are not found in an ancestral group. And a clade itself is an evolutionary branch in a cladogram, and it consists of ancestral species and all the descendants. Okay, so a clade consists of the ancestral species and all the descendants. So let me give you an example of a clade. That would be a monophyletic clade. Okay? That would be a monophyletic clade. Because it includes an ancestral species and all of its descendants. Okay? This, on the other hand, let me just undo that. This would not be a clade because it does not include all of the ancestors, or sorry, the descendants. Okay, this would be the ancestor, these would be the descendants, right? This is not a clade because it excludes those guys. Go back again. This is a clade because it includes the ancestor and the descendants. Okay, get the idea? Let's try some examples. So here we have a couple of different examples. Again, here is our just kind of arbitrary cladogram, and here is an example of a non-monophyletic group. Okay? Now, let's take a look over here. What over here we have is reptiles. Okay, now the reptiles, get a different color for this. The reptiles are right there. They are not monophyletic. Okay, now I want you to think about that. If this is the common ancestor of the reptiles, why are these reptiles not monophyletic? Well, here's the deal. If I was to do this, I'd be excluding this guy. These crocodiles are more closely related to these birds than they are to these other reptiles. So the group of reptiles is not monophyletic. It does not include all of the ancestors and the descendants. The descendants in this case would have to include the birds if it was to be monophyletic. Okay? And that happens because the crocodile is more closely related to birds than it is to snakes and lizards. Alright, so let's try a real life example and see how you guys do with that. Okay, so here we have some data, some molecular data of comparison between these different organisms. And what we're going to do is we're going to create a cladogram. We're going to construct it, if you will. And there are some different steps you have to go through, and there's some boxes in your booklet there that kind of help out with that. The first box, step number one, is that you have to sort homology from analogy. Okay, so homology, by definition, is a group of characteristics that are similar in structure and are shared ancestrally. Okay? So basically what that means is, let me give you an example. The bones in a human hand and the bones in a bat wing are very, very homologous. The structure of them, the number and types of bones are almost identical, even though my hand and a bat's wing have very different functions. Okay, so they're similar in structure, and we also come from a shared ancestor. In this case, we're both mammals, so we have a common ancestor somewhere in the past. Now, the greater number of homologous parts, the more closely related things are. Analogous structures are the opposite. They would have the same function, but the structure would be very different. For example, the wings of a bird and the wings of a butterfly. The wings of a bird would have all kinds of bones in them, where the wings of a butterfly wouldn't. They're very different, even though they're both used for flying. Okay, so the number of analogous structures has no bearing on how closely related they are, whereas the number of homologous structures does. Okay, so that's number one. Number two, you have to separate those two things. Number two, you have to identify the shared derived characteristics. For starters, the shared primitive characteristics, okay, those are not limited to the group being studied. For example, a backbone and mammals. Backbone is not limited to mammals. Therefore, I wouldn't necessarily use that to sort things in my cladogram. However, shared derived characteristics are evolutionary novelties that are unique to the clade. And therefore, you can use them. Example, hair and mammals. Hair is unique to mammals. Backbones are not. You would not use backbones to separate organisms in a clade from mammals, because they would all have it. Hair, on the other hand, you could use to separate mammals from other clades. The third thing is an outgroup comparison. An outgroup is a species that are closely related to the species being studied. An ingroup is the species being studied. You have to identify characteristics that you would like to study. You have to create a data matrix. This is a data matrix. And you have to then construct your cladogram. Okay, so here we have our data matrix. How are we going to then separate these organisms? Okay, so how do we read this, in other words? If I'm looking at the horse, okay, the horse is 0 separation from itself. Well, that makes sense. If I'm looking at, I'm going to use different colors for these. If I'm looking at the donkey is 1 separation from the horse. And if I'm looking at the whale is 5 separated from the horse. And if I'm looking at the chicken is 11 separation from the horse in terms of this particular protein. If I'm looking at the penguin is 13 separated from the horse. And if I'm looking at, I don't know, let's pick that one. The snake is 21 separated from the horse. So that's how far apart they are. That's how closely related they are. Okay, you can go the other way too. You can pick any one of these organisms and you can look and find the different things that you need to know. Okay? All right, so which two organisms, the thing I would do is I would put the two organisms that are least closely related on opposite ends. But I would also look at which organisms are the most closely related. So obviously the horse and the snake are the two opposites. So they would technically go here and here. But how do you know which one goes where? Well, let's look at the other end. Okay, so the snake. The snake is 13 and 21. That's 8 units away from each other. The horse and the donkey are 1 unit away from each other, and therefore, if you look at this picture, they're 1 unit away from each other. That little bit of space right there represents 1 unit. This big space here likely represents the 8 units that these two are separated. Therefore, the snake is going to go here. Okay, and I'm just using my colors. I don't want to write because I'll get all messy. I'm using my colors, and the horse would go there. Okay, the donkey would go there because it's the closest related to the horse. Next one we would have, what color did I use for the whale? Uh-oh, I lost my place. What color? Red for the whale. Alright, so red for the whale would go there. You get the idea? The next one will be the chicken. Come on, chicken, bark, bark. We get the penguin next, orange, and then last but not least we have the snake. Okay, so that, in a nutshell, is what phylogenetics are and systematics and how you would both interpret and draw a cladogram. Make sure you fill in your notes and bring any questions you might have to class.